Hello, everyone. I'm Dr. Joanne. Have you ever wondered what it would mean if we could rewrite the very code of life? But here's the deeper question. How would our society be impacted if gene editing technologies like CRISPR became the norm in human medicine? Today, we'll explore the ethical implications of CRISPR. Imagine that we can engineer and customize perfect humans who enhance both physical and mental traits, such as enhanced physical appearance, decreased susceptibility to non-communicable diseases, higher intelligence, or features such as different eye colors and increased height. What was once science fiction has become a reality with a technology called CRISPR. This technology gives us the ability to rewrite the very code of life. With this breakthrough technology, there are some ethical concerns that we must consider carefully. CRISPR has already demonstrated enormous therapeutic promise. This revolutionary technology is being used in human clinical trials, particularly to treat conditions such as blood diseases, cancer, and HIV. Scientists in Philadelphia, including Dr. Khalili and his colleagues, have shown that they could use CRISPR to remove the DNA of an integrated HIV from infected human cells. The main goal of this biotechnology is to advance medical treatments to prevent genetic diseases. It could offer substantial therapeutic benefits, but it also brings serious ethical problems, since future generations cannot consent to gene changes. The majority of countries have discouraged or prohibited heritable human genome editing, and some countries allow specified exceptions, but these must not result in a live birth because of ethical and safety concerns. In November 2018, He Jiankui, a Chinese scientist who sparked global controversy by creating the world's first gene-edited children, using CRISPR in an attempt to make them immune to HIV, was sentenced to 3 years in prison because he was guilty of the illegal practice of medicine. Although CRISPR is promising, there are many effective methods to prevent HIV in healthy individuals, such as practicing protected sex and using available treatments if one is diagnosed with HIV. Yet, gene editing has occurred during in vitro fertilization procedures, and this experiment exposes healthy children to the risks of gene editing with long-term implications for uncertain benefits. On the other hand, the world's first approved CRISPR-based cell therapy is being used widely in wealthy Western countries. In those countries, genetic therapies can cost upwards of $2 million for a one-time treatment, according to Rueda, de Miguel Berrien, and Montoliu, 2024. This kind of elective procedure is only going to be accessible to people who can afford it, which could deepen social, economic, and even racial divisions. Although CRISPR-Cas9 technology may offer unlimited benefits, it is difficult to imagine a world where being genetically modified is the norm, and those who cannot afford it would likely suffer mentally and even physically. Another concern is that gene editing isn't 100% accurate and that it interferes with nature. To illustrate this point, Kozubek expresses concerns about the future impact of gene editing, particularly its potential to interrupt natural evolutionary processes and create unpredictable biological risks. CRISPR should not mirror a dystopian society of genetic classes. Instead, it should serve as a means of promoting accessible and equitable healthcare and education. In conclusion, gene editing technology is at the threshold of drastically affecting humanity. It brings enormous promise in the form of reducing or eliminating many diseases. However, it could also shrink the human gene pool, turn human traits into consumer choices, challenge human dignity, widen socioeconomic divisions, and have unprecedented consequences for future generations.